Okay, so we're doing Lab Test 3 review, and we're going to start with human genetics. And this first question, of course, is just referencing humans. In humans, where are autosomal characteristics carried? I'll help you out. It's chromosomes 1 through 22. You may remember we have 23 pairs of chromosomes, and the first 22 pairs are what we call autosomes. They're just carrying traits that you get two different alleles for. So we call those just autosomes. Whereas the sex-linked traits are on the 23rd pair. And you may remember that the 23rd pair is the X and the Y chromosome for males, or the X chromosomes only for females. Now, just to remind you, because it has been so many weeks ago since we talked about this, would you glance at page 123? Page 123 has a karyotype at the top, and you can see what I'm talking about. Chromosome pairs 1 through 22, and then the 23rd pair of chromosomes are always over on the right-hand side at the bottom. Okay, on question 1. What chromosomal abnormality is associated with Downs? Yeah, extra chromosome of the 21st. That's good. Hey, Charles, you want to get a review sheet? So we actually call that one trisomy or trisomy 21 to indicate that it's 3 21st chromosomes. Make sure you could spot that on a karyotype. So you just look at the 21st pair and see if there's 2 chromosomes there. If you see 3 chromosomes there, you know that that child would have Downs. Okay, how can you tell the sex of an individual in a karyotype? That's right. So you're going to look at the 23rd pair, and let's say you see 2 nice-sized chromosomes there, and they're about the same size and shape at the 23rd pair. What would you say the baby is? That's a girl. Okay, what if you saw a big one and then a really little one? That one's a boy, and that little one's called the Y chromosome. Of course, they're showing you on page 123 a little boy. Page 123, top of the page. Everybody can tell that's a boy? Jacob, you're good on that? Okay, good. What chromosomal abnormalities are associated with Turners? Does anybody remember Turners? Okay, I'll help you out. Page 125. They only have one X chromosome. It's good, Victoria. They only have one X chromosome. So how come I put an O there? Yeah, that really stands for 0. It's a placeholder. Okay, how about a lady that has poly X? What would that be? 3 X's. And again, if you don't remember this, it's page 125 in our lab manual. Okay, then a gentleman with Klinefelters, what would that be? XXY, good. And lastly, a gentleman with Jacobs. XYY. Good. In class, on the test, I'm going to give you karyotypes, just like on page 125, and you have to figure out what disorder the baby has. We practiced that on page 125 in the middle of the page. Gabe?
So, will you ever play what gender has to have a certain type of abnormal?
No.
No, you're just saying
No.
XXY, what is that?
XXY. Yeah, I could do it that way, but I'll probably do it because I think I did it that way on a lecture test. So then I'm going to change it up on you and do it visually by giving you a karyotype.
Okay.
And I'm just going to give you the karyotype, and then we'll have choices, and you pick the right choice. So you got to be able to study the chromosomes and figure it out. Now there's only 5 things it could be: Downs, Turners, Poly-X, Klinefelters, Jacobs.
And the karyotype, it's never going to give you the 23rd, like it doesn't say 23, it just shows them.
It just shows them over there on the corner, yeah.
What is that, the 21st chromosome have to have for it to be considered down?
3 of them.
3 X's?
Well, 3 chromosomes, yeah. And I agree that they all look like little X's. Yeah. Okay, now we're going to switch gears from looking at whole chromosomes to looking at individual genes on chromosomes. And that's mostly what we worked on because chromosome abnormality, that's common. But what's more common is to inherit genes from your parents that could cause some kind of problem. This is actually more common. So just to review with you some genetics. So I have number 5, if a person is homozygous dominant, I went ahead and wrote their genotype there. Or if the person is heterozygous, what characteristic will be shown in their phenotype? Will they look dominant or will they look recessive? They're going to look dominant, okay. Okay, how about if an individual is homozygous recessive? What trait will show in their phenotype? Recessive. Question 6. You would expect the dominant trait to be the most common trait in a population. Is this always true? No. Like, would you guys agree with me that blue eyes is recessive? Okay, do you think there are places in the world where I can go where blue-eyed humans are more common than brown-eyed? Yes. Okay, so why? It's strong. I've got blue eyes. I don't feel strong. Okay. Recessive what? Okay, so the people who started that group had blue eyes?
Not always.
Could be the founder effect. That's called the founder effect, right? Where everybody in the population looks like the founders because the founders started the population. Okay, or it could be that blue eyes would be favored. I don't know when that would be the case. I can't think of an environment that would favor blue eyes. I really can't. But it could be that blue eyes would be favored. That would be called natural selection, right? So it could be things that have to do with evolution. You know, I really should look into that. There surely is some advantage to blue eyes. I just don't know what it is. But okay, number 6. Okay, number 7, if given information about which trait is dominant and which is recessive, be able to work genetics problems. Well, I wanted to remind you that there's going to be some genetics problems on the test, so I want you to go back and review a few. Page 127, would you just flip over there? We did question number 1. It's towards the bottom of the page about Nancy and everybody in her family. And don't forget, I will always tell you what's dominant, what's recessive. And then on the next page, we did a problem about Joe and about Henry. So I would go back and review those questions and just make sure that you can work those genetics problems. They're real easy. It's just been a little while since you worked on them. So just go back and review those. So I came up with a genetics problem. It's not on your review sheet. It's only up on the screen, just to make sure that everybody can do it. I have 2 people with widow's peaks, have 3 children. Now, widow's peaks is dominant. Having a widow's peak, do you remember what that is? It's what I got. That's right. Yeah, I see a couple of you that have it. That's dominant. And if you have the straight across hairline, then that's recessive. Okay? All right, so I have these 2 people. They have 3 children. 2 of their children end up with widow's peak, and 1 of their children has a straight hairline. What are the genotypes for each? So we got to figure this out. Now, I wrote a key at the top just to remind you. Capital W is widow's peak, and lowercase w is straight. Let's start with the parents. They both had widow's peak. That's good, Victoria. Big W, little w, big W, little w. Now, how did Victoria know that the parents both had to be heterozygous? Because one of their children had a straight hairline. Okay, good. All right, now on the children. Let's do the straight hairline first. He's the easy kid. What would be his genotype? Yeah, the little w, little w. But what about those other 2 kids that had the widow's peak? Okay, anything else? Yeah, both of those are a possibility. So based on what we know, it could be either one of those. But okay, on that, it's just real simple genetics problem. Okay. This one's true or false. Sex chromosomes carry only genes that relate to the sexual characteristic or sexual functions of an individual. Really?
False.
This is false. So X chromosomes have over 3,000 genes on them, and they don't all have to do with our sexuality. Can you give me a gene on the X chromosome that has nothing to do with sexuality? Guess not. Yeah, color blindness. Has nothing to do with your sexuality. Here's another one in case you maybe were thinking this. Hemophilia, that's where people can't have their blood clot very well. That has nothing to do with their sexuality, but it's on the X chromosome. The most common cause of muscular dystrophy is on the X chromosome. Again, another example of a gene that has nothing to do with a person's sexuality, but it's on the sex chromosome. Okay. So speaking of the X chromosome, all the traits on the X chromosome are called X-linked traits. How are X-linked traits inherited differently in males and in females? Like who tends to show X-linked traits like hemophilia or color blindness? Males. Mm-hmm. Okay, so then what about females? They don't. They hide it. They're carriers. Very good. Okay, so color blindness is an X-linked recessive disorder. I just wanted to remind you the day that we were working on this activity, I showed you a couple colorblind tests. This one, I think you'll remember, is really good for younger children that haven't really learned symbols, numbers, letters, things like that. Or a person that maybe isn't fluent in the language that the administrator is using. But this is the one that's the more common one where I think we spent a little bit of time looking at all of these, coming up with what they showed us. So that was kind of cool. That's a different colorblind test. But I think you'll agree, you meet more people that are men that are colorblind and hardly any women that are colorblind. And again, that would indicate it's on the X chromosome. Is okay on that? Super. Okay, pedigrees. So in our lab manual, would you turn with me to page 131? That's pedigrees. Page 131. So a female. What is the symbol for a female on a pedigree? Circle. And males are? And if you show the disorder, what do? Color it in. Okay, now you're going to have to be able to tell me how a trait is inherited. And you have three choices at the top of page 131. Autosomal dominant, autosomal recessive, and X-linked recessive. So you want to go back and review those clues. So I made up a pedigree just for fun. It's up here on the screen. Everybody glance at my pedigree. And the first thing you want to do when you're doing a pedigree is figure out the inheritance pattern, whether it's autosomal dominant, autosomal recessive, or X-linked recessive. Which one is this? Autosomal recessive. It's clearly recessive because neither original parent has it and only one of their three children does. But then why wasn't it, and I agree with you, but why wasn't it X-linked? It's in males and females. Yeah, you wouldn't expect that if it was X-linked. Okay, we're just going to use the letter A to stand for the trait. And let's try some genotypes here. Class, let us do generation 1, individual 1. Go ahead. I'll take any nominations for the genotype of generation 1, individual 1, using big A, little a. Now, how did you know it was big A, little a? Because one of their children have it. That's right. So clearly this one is also the same. It's the only way you could have two unaffected parents and then an affected child. Everybody okay with that? Okay, let's try that child. Generation 2, individual 4. Little a, little a. Okay, let's try the lady he married. Generation 2, individual 5. Big A, little a. That's good. Let's do both of their daughters. Generation 3, individual 4.
Big A, little a.
And the boy. That's good. Everybody okay on that?
They're genotypes, right?
That what I put up here in pink are genotypes. Sir?
So number 4 could not be little a because big A because one of the parents has it. Is that right?
So you're talking about generation 3, individual 4?
Yes, ma'am.
Okay, so that one couldn't possibly be big A because of that dad right there having 2 little A's. Because dads get to equally contribute to their children just like their moms, okay? I would recommend you looking at spending a bit of time on the 2 pedigrees on page 131. Those are really good pedigrees too. Pedigrees will be on your test. And don't forget, step number 1 is to figure out what the inheritance pattern is. Then once you figure that out, then you can do the genotypes. Okay, I'm going to put a blood type problem on the test. I have Susan has blood type A, Jack is AB. What are the possible blood types of their children? Okay, well, I got this table that's from our lecture notes. I just want to remind you that when we say a person's blood type, we're really saying their phenotype. And that if you're blood type A, you could be homozygous A or heterozygous A. If you're blood type B, you could be homozygous B or heterozygous B. But if you're AB, you're only AB. And if you're O, you're just definitely OO. So looking up there, I would say somebody's recessive. Something up there is recessive. What is it? O is recessive. And it looks to me like A is dominant and B is also dominant. We'd say they're co-dominant. A and B are co-dominant. Okay, so let's go back to our problem. And our problem was Susan's blood type A and Jack is AB. Susan is my issue. Aunt Susan, should I use big A, or big A-O?
Big A.
I'm going to use big A-O. So I'm going to work the problem. And this is Susan, big A-O. And there's Steve. I think his name was Steve. No, it's Jack. I'm sorry. I have Jack, and he's across the top. And so we can go ahead and fill in our squares. So this is going to be one of those choose all that apply. Okay, could Susan and Jack have a baby blood type A?
Yes.
B?
Yes.
AB?
Yes.
O?
No.
Make up your mind.
No.
It's no. Don't pick O for this problem because you couldn't do it. To have a baby O, doesn't the baby have to be O? Are there any babies up there? O? Could a person with blood type AB have a child that's O?
If they mate with someone who's O.
No. Listen, here's the question again. Could a person whose blood type AB have a baby that's O?
No.
No.
So both parents have to be O.
There has to be an O hiding in there on both parents. AO or BO. Exactly, Charles. Yes.
My mom was O. She used to be OO. And then my dad, I'm not sure, so, but I'm A.
You're A. So your dad's got an A in there somewhere. He's either A or AB. You should ask him. Maybe he'll know. But lots of people don't know their blood types. If you donate blood, they will send you a card that has your blood type on it. Okay, well, we're going to leave that activity, which was exercise 10, human genetics, and move to exercise 11, DNA biology and technology. And I do want to tell you that the artwork on page 136 is pretty good. 136. And your lab manual has some pretty good artwork. I can't find any errors on it. You know those DNA drawings, sometimes there's errors, but I can't find anything wrong with this one. Okay, so the first thing we have to do is do these three words. And so these three words, every time I see them together like that, I think of this. I know I've drawn this for you a couple, 3 times. And so the three words that I want to put on this picture are replication, transcription, and translation. So I'm going to start with the first red arrow that shows DNA is used to make more DNA. Would you call that replication, transcription, or translation? That one's replication. Okay, then the next arrow shows me that you can take DNA and make RNA from it. What would that be? That one's transcription. Good. And the last one shows that I can take RNA and make protein from that. Translation, yes. And we're going to practice those words as we continue. Okay, question 2, the 3 parts of a DNA nucleotide. Well, I'm going to draw one. I got a P with a circle around it. Then I have this thing that kind of looks like a house. And that O at the top of the house is oxygen. Inside the house, I have the letter S. And then I have a square with a B in it. Okay, let's talk about with the P that has a circle around it. What is the P with the circle around it? What is that? Good. Okay, then I got this thing that kind of looks like a house. Okay, you may not have remembered, but sugar's not going to be a choice.
Deoxyribonucleic acid.
Okay, when would the sugar be ribose? For RNA. That's good, Eric. For RNA. But this question said we're doing DNA, so it's deoxyribose. Okay, so I just want to remind you, sugar is not going to be a choice. Okay, how about the B with the box around it? Those are the nitrogenous bases. Now we're doing DNA. What are my choices? T, okay. A, C, and G. That's good. Because this is DNA, so those are my choices. But okay on the 3 parts of a nucleotide? Okay, one more time. Would you look at page 136? I think you'll agree on page 136 that DNA looks like a ladder. Yes? Let me show you. You built something like this before. I'm going to talk about this model in just a second. But don't you agree it looks like a ladder? Okay, so thinking of DNA, it looks like a ladder. What parts make up the sides of the ladder? No. Look at the picture, page 136. The sides of the ladder. Phosphate and the sugar. What's the name of that sugar? Deoxyribose. Everybody see the sides are made out of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. It's holding it all together. It's structural. Oh, look at the cute turtles back there just getting sun. You see them looking around? They're trying to learn about DNA. Okay, what are rungs of a ladder? Rungs? Rungs are steps. Okay, maybe you don't know that word. So the steps of the ladder. How about that? What molecules make up the steps of the DNA ladder? The A and the T, the C and the G. And what holds them together, the A and the T? Good, Jason. Hydrogen bonds. How many between A and T? Look. 2. And how many hydrogen bonds between C and G? Look. 3. You guys are guessing. And again, the artwork on page 136, pretty good. So I took a picture of this and put it up here, and I drew white arrows to the things I think you should be able to tell me. Now, please agree with me, this is a picture of DNA. Okay, so everybody's fine with that this is DNA, so I'll write that down for us. This is DNA. How would we know? Because it's a ladder shape. It's got 2 sides. It looks like a ladder. That's good, Charles. All right, let's start with, I'm going to put a number 1 right here by this arrow. I want to know what that white tube is that the arrow is pointing at. Phosphate. That's good.
This picture will be on the
This will be on the test. It won't be a picture. It'll be this.
well, like that actual picture, just like.
This is a picture of this. So I don't want you to think I'm putting a picture on the test.
What I'm asking is that's going to be on the video.
We're recording the video as we speak, and your voice is on it right now.
I thought it was like.
Look at 29 minutes, and there you're going to be right there.
Like and subscribe.
Okay, going on down. Number 2. Number 2. How about number 2? Okay, sugar, thank you. Sugar's not going to be on the test, but the word deoxyribose is. You'll be fine. Even if you think sugar while you're taking the test, you'll look, you won't find the word sugar, then you'll remember, oh yeah, and you'll pick the right one. Okay, and I got one more way up here at the top. Now, I don't see if you know if you can tell, but it's those white rods that are holding the steps together. Hydrogen bonds. Good job. Okay, now, how about this blue thing right here?
That is a base.
It is going to be a base. Now, let's pretend. Okay, let's pretend it's C. Then what would the black one be? G. That's good. I can't write on that very well. That's a G. It's a bad G. Okay, how about if the red one is A and T? So you should be able to do complementary base pairing for DNA. So I wanted to make sure that you could. So I made up a just pretend sequence here. This goes with number 3 on our list of questions, but this little pretend sequence, I just put it on here. So I'm going to go ahead and let you tell me what the answer is. So go ahead and tell me first what number I would put down here. 5. Okay, go ahead. Good. So everybody's fine on doing complementary base pairing. Great. Okay, we're going to make a little table here comparing DNA and RNA. And guys, I'd like to find 3 differences between DNA and RNA. I'd like 3 differences. Go ahead. That's good. So DNA has deoxyribose. And RNA has the sugar ribose. DNA has thymine, or the letter T, and uracil, or the letter U, for RNA. Monica's right. DNA is double-stranded. I'm running out of room. So you could put stranded on there. And RNA is single-stranded. So we have the difference in the sugar, the difference in the base, and the difference in the overall structure. DNA double-stranded, RNA single-stranded. Everybody okay with that? Thymine. You can just put T and U. Okay, so I took a picture of something else. We'll hold it up. RNA. What kind of RNA?
mRNA.
It is. Who told me that? Good job. That's mRNA. What's M stand for? Messenger. Okay, we've got to do some parts. Let's start right here with this white tube.
Is that number 5?
Just a picture. What's this one right here? The white tube.
That's the phosphate.
Sugar. Wait, no.
The white is phosphate.
Okay, let's do this blue pentagon next. Okay, now I have my arrow on a red tube. I could also put the arrow on the blue tube or the arrow on the black tube. What are those? Huh? Bases, good. What bases could they be? AU. CG. Okay, on my picture. And remember, the picture's not on the test, but this real thing will be, okay? Super. Okay, here's some vocabulary that kind of takes us back. And I think people forget these words, so I want to show you where we used them before. Check out page 143 at the top of the page in your lab manual, page 143. See where it says mRNA codons. So how many letters in a codon? 3. So a codon is 3 bases. And you always have codons on a molecule that's called mRNA. Does anybody remember what a codon codes for? The parts of proteins, what are those called? Starts with an A. Amino. Code for one amino acid. I use an abbreviation, amino acid. Okay, now to know how to read the message and put the amino acids in the right order, you got to have a particular organelle. But remember what organelle is associated with making proteins. Starts with an R. Okay, I've given you a piece of DNA up here. How do you know that's DNA? As a thymine. And I'm asking you to transcribe it. So what am I asking you to make? No, you don't need to use a chart yet. That's translate. I want you to transcribe it. What am I asking you to make? RNA. So go ahead and get me started. No. There. Go ahead. Who was that? Well done. He's got his transcription down. Okay, so one more time. If I ask you to transcribe something, I want you to make RNA. Got to know what that word means. So notice I didn't give you any other prompt. I just used the word transcribe. It's okay on that. Now, guys, we did practice that on page 141. We practiced transcription at the bottom of page 141. One other quick comment. We did have some nice artwork on page 141 where you have the double-stranded DNA pulled apart and the single-stranded messenger RNA is being made. There's some pretty nice artwork there also. All right, I took a picture. Man, there's so much going on up there. Just see if you can help me figure out the 3 major parts up here. I'm going to start with this big blue thing. I'll call it number 1. What's that big blue thing? That's a ribosome. And look, it's attached to this long white thing that has bases. I'll call that number 2. What is it? mRNA. That's mRNA. Okay, and then down here on the bottom, I have 3 of these. Here, I want to circle one. I have 3 of these. I bracketed it here. That's tRNA. Okay, so think hard. What process did I take a picture of? Transcription? Translation. This one's translation because we're making an amino acid sequence and we're using a ribosome, messenger RNA, and transfer RNA. This one's translation. Translation. Okay, now what I have the box around is a tRNA molecule. I took a bigger picture of that. Okay, it's tRNA. Okay. The tRNA is actually the blue part right there. But there's some other parts here that I have white arrows on. Let's start at the top. Does anybody know what you call these 3 bases on a tRNA? Anticodon. That's good. Okay, that's so loud. I can't believe that. What if they lived next door to you and they went to work at 5 in the morning? I don't know. It's just loud. It's just a hot rod. Yeah. I digress. Number 2. Number 2 is this black thing hanging off the bottom of the tRNA. Amino acid. That's good. I'm just going to abbreviate again, amino acid. Okay, so again, this picture is a tRNA. It's just enlarged, showing you the parts of the tRNA. It's okay on that. Now, keep that in mind. We go to the next question. Keep this picture in mind for the next question. What 2 components necessary for translation are carried by tRNA? The anticodon. What else? Amino acid. What is an anticodon? Okay, so it's kind of hard to put into words, so I want to show you where you did some anticodons that might make more sense to you. The top of page 143. At the top of page 143, I gave you codons, and then you gave me anticodons. So it looks like an anticodon is 3 bases. Aren't they always in 3s? Yes? And they're found on tRNA. You know, mRNA has codons. tRNA has anticodons. Now, I'm going to give you some codons on the test, and you have to give me the anticodon. So let's practice that. See, I gave you 2 codons up here, and then you give me the anticodons. Everybody see my practice? All right, go ahead. The first one. AU who? Okay. I thought I heard somebody say T. That's why it hangs out with you. But okay, I'm doing anticodons. Okay, great. Would you find your genetic code? Now, on the test, I'm going to have the genetic code for you to use. You don't have to worry about supplying your own. I have the genetic code as part of the test, just like usual. But if you would, get that out. Why? Well, everybody look at this next one. How do I know I need the genetic code? Translate. I got to translate. Now let's translate this using our genetic code, and like usual, we're going to use the first 3 letters of the amino acid as an abbreviation. I'm going to get us started with AUG. So on my code, is there anyone who needs a code? Charles, come on up. AUG on my code. It's over on the left-hand side, and I see that it's not only the start codon, because everybody knows it is, it also codes for methionine. So I put MET as the first 3 letters of methionine. Okay, how about CGC? What'd you get for that? That's good. Arginine. ARG, arginine. Okay, the third one is UAU. What'd you get for that one? Tyrosine. That's good. And then we're doing UAG. Perfect. Is okay on that? Just refresh your memory on how to use the code. Good. All right, so we had this other handout that was called electrophoresis. Could you get that out, please? Electrophoresis. And I hope that you still have it because you had lots of data that day, but also on the back, you drew a test tube and labeled some parts on the back, and that's what I'd like to look at right now is the back of the handout that's called electrophoresis. Does everybody have that? Anybody need one? Okay. All right, so you guys are going to have to get some notes maybe from Lewis. Okay, so on the back, if you were here that day, we did in handwriting during the DNA isolation, what role did each play? And we had banana cell extract. What was the purpose of the banana cell extract?
For DNA.
Okay, then we had this detergent called SDS. What was the purpose? So that emulsified the banana's membranes. Just getting this off of the back of your sheet there. And then how about the ethanol? What was it for? Yeah, extracted and attracted the DNA. So made the DNA rise to the top and separated it away from the rest of the cellular contents. Is everybody okay on the DNA extraction? Okay, now let's keep that sheet on electrophoresis, but this time turn to the front of it. I think in the very first sentence we can find what gel electrophoresis is used for.
Separate molecules.
Very good. Gabe said to separate molecules. Upon what basis does it separate molecules? Would you say size and charge?
Yes, ma'am.
Now this last question is so easy, people just don't even think about it. I have what makes the molecules move. You had to plug it in and turn it on. What was it? Electricity. That's what puts the electro in electrophoresis, is the electricity. If you don't plug it in and turn it on, nothing's going to happen. Okay, this looks really long, but have no fear of it. So, in the middle of the page, there's this paragraph. I'm going to read it to you. It says, each protein has an isoelectric point, the pH at which the protein is not charged at all. In solutions with a pH higher than the isoelectric point, a protein becomes more and more negatively charged. In pH solutions lower than the isoelectric point, a protein becomes more and more positively charged. And so then what we did, guys, is I gave you the isoelectric points and different pHs, and then we guessed what the charge would be. So you just would go back and review that. But I have a little pretend gel up here. Look at my pretend gel. Let's say that these are pigments. Which pigments, I just numbered them, which pigments would be positively charged? Heard 1 and 5. Does everybody agree? Okay, which pigments would be negatively charged? 2, 3, 4. Okay, now look at 2, 3, 4. Looking at 2, 3, 4, I also want you to notice the pH. Did you see it at the very bottom? So it's neutral. Okay, which of these, 2, 3, or 4, has an isoelectric point closest to neutral? 4. And which one would be the furthest away? 2. Okay, so that's what I mean by this last sentence on number 11. Be able to determine the distance a protein will travel relative to other proteins.
You're gonna give us one?
Something like that. Yes, ma'am.
I wasn't here when that happened, but the 1 in 5 is positive because it's attracted to the negative.
Perfect.
Sounds like it.
So, I mean, I don't know if this makes sense, but would 2 be the lowest pH out of all of them?
You mean the isoelectric point would be the most different from 7 out of those 3. 1 and 5, you can tell also, it's closer, but 1's isoelectric point is closer to 7 than 5's. But they're kind of close. Yeah. All right. That takes us to the back. So our last unit that we've been working on together is evolution. And we've defined evolution actually several times. Use the same definition every time. So when you're studying a population, if you see it changing, we would say it has evolved, or that it is evolving. So evolution is the genetic change in populations over time. Again, we've written this definition several times in our lecture notes and in our lab notes. How does life explain the unity? How does evolution explain the unity of life? Like, how come all cells have DNA and mRNA and rRNA and tRNA and they all use the same genetic code and they all use ATP? How come? Common ancestry. Good. Okay, how does evolution explain the diversity of life? Like, how come there are so many different kinds of birds? Or how come there are so many different kinds of flowering plants? How come?
Migration?
Evolution for what? Like, why? Why did it evolve?
The change of
Change in what?
environment.
Very good. Yeah, if all the Earth was the same environment, we wouldn't see diversity in life. But as you know, there's so many different environments. So many. You know, just living in the crack between 2 pieces of sidewalk is an environment. Okay? It's in a microenvironment, and it takes a special organism to live there, but that's an environment. All right. Page 151. Sorry, page 150. I think you already wrote these down. Let me know if that's true. What does Paleozoic mean? Good. How about Mesozoic? Huh? Middle? Okay.
Page 151, you said?
I think it's 150. How about Cenozoic? Recent. Okay, now these words, you have to know what they mean, and then you have to know which fossils that we studied are from each of these 3 eras. And there are 12 of these fossils. I do have pictures of these fossils on Blackboard. I think it's like, I don't know, under course content somewhere. Have you guys seen them? It's a PowerPoint on the pictures of them. It's under course content. I don't know what's past that, but it's under course content. Look around. You can find them. But the fossils are also in those red boxes over there. I'm going to look at them. Okay, homologous. Page 154. What's homologous mean? Same structure or same function?
Same function, same structure.
And this is on page 154. Parts of organisms are said to be homologous if they exhibit similar basic structures and origins. Okay, the companion word for that is analogous. What's analogous mean? Same function, that's right. In your opinion, does homologous or does analogous represent a closer evolutionary relationship? Structure or function would show evolution. Structure or function? Structure. Guys, on page 155, I want to point out that we had a comparison of frogs, birds, bats, cats, humans, and lizards to the amphibian ancestor in the center of page 155. Would you say that the humerus on each of those forelimbs would be homologous or analogous? Same structure or same function? Same structure. So is that homologous or analogous? Homologous. So everything on page 155 is homologous. Homologous. Okay, next page. I'm on page 156. We compared human skeletons to chimpanzees. Now, from here down, come up with one reason why we're different. Okay, hips and femurs are different, I agree, but why?
Because the way we walk.
We're standing on 2 feet, and so we have to balance over our hips, whereas they're still using 4, so they don't have that balance. They're thrust forward.
Did you say that babies were like
They're shaped like that.
first and then...
They sure are. They wobble all right. Okay. No, I was holding my granddaughter last night. She's 3.5 months, and I was kind of starting to let go of her, and she's wobbling all over the place. Made her real mad. Grab her again. Yeah. Yes.
Like you said, like normally the backbone is like at a certain age, that's when they can just sit upright and go like this.
Yeah. So the hold the head when they get this curve in the neck, and then when they get the curve down, you know how we have a curve on right above our buttocks? That's when they can sit up. Yeah.
What if they just start turning their heads like on their own?
Well, holding your head and turning your head are two different things. Yeah. So it's when they can hold their head. Yeah.
They can turn their head.
Yeah, they can turn their head at birth so they can find the breast, but holding it by themselves, that's pretty difficult. So she's still working on that. All right, number 6. So the next page, page 158, I've got a chimp skull versus a human skull. We're looking for the differences there. There are many, but we want to know why. And I think there's 2 reasons why chimp skulls look different than human skulls. I need 2 reasons. Okay, so brain size would explain all the differences you see up in the cranial region. We have a larger brain. But what about at the jaw? Why is the jaw different? Diet. Good. So the difference in brain size and the difference in diet would be the 2 main reasons that the chimp and the human skulls look different. Speaking of skulls, I got 12 skulls in the back. I'm going to ask you to identify the animals. Now, I will be fair. I'm not going to put the skunk on there and put the skunk and possum as choices because the skunk and the possum are so similar. Okay, so I promise I won't do that, but you need to make sure you know the skulls. There's a PowerPoint on the skulls also on Blackboard. And then another thing is that their teeth. I remember telling you that I want you to know what kind of diet they have, and you can tell by looking at their teeth. And your choices are going to be herbivore, carnivore, and omnivore. But okay, on the skulls.
Because we're
Omnivores will be both. So it's like ours where we have the molars in the back, but they're a little ground down. So, you know, if you found let's just say you found a jaw and every tooth in the jaw was sharp. That's a carnivore. How about if you found a jaw and these back teeth were ground down? Herbivore. Okay, and then, you know, if you had something that looks kind of like a human, then that's going to be an omnivore because we're in the middle. Okay. All right, comparative embryology, page 159. So I remember in lecture one day, we had these 4 embryo pictures up on the screen, and we were supposed to figure out which one was a lizard, which one was a bird, which one was a pig, which one was a human. And to me, the hardest one was figuring out the pig and the human because they looked so much alike. Okay, they all looked alike, but the pig and the human looked a lot alike. Why would a pig embryo look more like a human embryo than, say, a chick embryo? Because we're both mammals, whereas chicks are birds. Okay, so the more similarities you see in the embryos, the more closely related they are. So I have, how does comparative embryology help determine relationships between organisms? More similarities, more closely related. Is that okay?
Like that same picture again, or just ask us a question about this?
Well, I could give you the picture that's on page 159. Although the only thing wrong with that picture on page 159 is they don't have the eyes right on that bird, do they? Yeah. How about this question? Would you expect a human embryo to be more like a chick embryo or more like a pig embryo?
Pig.
And then what was the reason why?
Because it's mammal.
You're a mammal, a pig's a mammal, should be pretty similar.
What is a dolphin?
Mammal. Okay, question 8. I can give you a page number on this, but you guys know this. I need some biochemical molecules that all organisms have in common, and I'm going to get us started. DNA. Okay, give me some more. RNA. All 3 kinds. What else? Good. Ooh, I want to throw an enzyme up there for fun. ATP synthase. What's ATP synthase?
It's an
Enzyme that makes ATP. That's good. So there's bunches you could come up with. You guys know that all cells use the same genetic code, which was this. Okay, so there's just lots and lots of chemical similarities between cells. How come? How come there's so many chemical similarities between all cells? Common what? Okay. the other day, we had these on the table. And we put rabbit serum into the wells. Then we added human serum. And then we pretended 3 weeks were going by. Yeah. And then we found, like this little bottle says serum number 3. So I put serum number 3 in well number 3, and I looked for white precipitate to form. Now, we had used the rabbit as something to make antibodies, and we had injected human into the rabbit so it would make antibodies specifically for human protein. So let me show you a picture of that, page 161. So if the rabbit antibody recognized the protein as human, it would grab a hold of it. It'd be cloudy. If the rabbit didn't recognize the protein, it wouldn't grab it, and you wouldn't get cloudy. So here's the question. If you're looking for something really closely related to humans, would you look for a lot of cloudiness or not very much? A lot of cloudiness. You look for a lot of cloudiness. And of course, if I asked you to pick the species that wasn't very closely related, you would look for not too cloudy.
Wait, one more time, if it's clouded, what?
It's closely related to humans. Okay, lastly, on the back of your fossil page, there's a little activity we did there that goes with question number 10. Be able to use the cytochrome C protein clock to determine the degree of relatedness. If I were you, I would just rework questions 1, 2, 3 at the bottom. We did it as a class. We even did some clicker questions with this chart. Seems like you did pretty well on it, but be able to use this chart on the lab test. Everybody's okay on that? Does everybody have that chart? Yes? Okay. All right, the last experiment was mechanisms of evolution, and the first thing is just a lot of vocabulary, guys. And I'm not going to use your time on this vocabulary, but I'm going to tell you where to find it all. Okay, let's play some multiple choice on the agents of evolutionary change. Look at the top of page 166. This is really done well because not only is it multiple choice already, A, B, C, D, E, but they did different colors too. How nice. So, which agent of evolutionary change is basically migration? Alleles moving from one population to another. Gene flow. Everybody find gene flow. Which agent of evolutionary change is the ultimate source of new alleles? Where do new traits come from? Mutation. Which agent of evolutionary change will get you a population better adapted to the environment? That one's natural selection. That's good. Which evolutionary change may be due to inbreeding? Non-random mating. Inbreeding is an example of non-random mating. Okay, which agent of evolutionary change is just random and often found in small populations? Genetic drift. Okay, so there's your evolutionary change up there at the top, but they're all topics that we kind of discussed in lecture. Okay, but they're all on that page. Okay, Hardy-Weinberg has been just a ton of fun. I like it too. It's at the top of page 167. P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. So the first thing is, what's the purpose of this equation? Besides your teacher making you do math, does anybody know what the purpose of Hardy-Weinberg is?
To calculate the gene pool.
Calculate the gene pool. Why would you want to know it?
To know about the
Why would you want to know about the population?
To see where ahead, like if it'll survive or if it'll.
To see if it changes, right? Aren't we in the unit of evolution? Yes? What is evolution? Change in the genetics of a population. Okay, so I'm going to write that the purpose of Hardy-Weinberg is to be the baseline for evolutionary comparison.
Do we have a chart for that?
I don't know if we have a chart for that.
We did something.
Yeah, we're going to work a problem on that. So, you know, guys, I'm just going to give you an example. I want you, as a young adult, to go to the doctor and get your blood tested for cholesterol. And then, maybe every 5 or 10 years, you're going to go back to the doctor and get your blood tested for cholesterol. Now, why do I want you to have it done when you're young? So you can see the change, okay? And I don't know, some of you may already have problems. I don't even know, okay? But that way we know your baseline. So that in the future, if it changes, we'll know that it changes. So that's the purpose of Hardy-Weinberg, is to figure out the allele frequencies now so that in the future, like Charles said, so in the future you'll know if it's changed.
Did we use a graph or something?
We used an equation. We're going to do it some more in a minute. Okay. In the absence of evolution, so nothing's causing evolution, how would you expect gene frequencies of a large population to change over time? Would they? If there's no evolution?
No.
They wouldn't. There would be no change. Why would there be no change? Because there's no evolution. Isn't evolution change? So I'm saying that there's no evolution, so there would be no change. And that's what Hardy-Weinberg predicts. That they predict that if there's nothing that will cause evolution, a population's not going to change. I love question 3. It's the whole idea of why we start with Q squared when we do Hardy-Weinberg. True or false? The frequency of the recessive phenotype, you know, like how many people you can count in a room that have blue eyes, equals the frequency of homozygous recessive genotype, or percent little a little a. So if I count how many people in this room are recessive, that's not percent homozygous recessive. You count how many people are recessive, isn't that the percent homozygous recessive? Sure it is. That's true. And that's Q squared. That's what they're talking about right there, is what we call Q squared. How many individuals in the population are recessive? That's Q squared. Number 3 is true. Try number 4. True or false? The frequency of the dominant phenotype in a population equals the frequency of the homozygous dominant genotype. Say it again. Yeah, it's P squared. So if I count how many people are dominant in a room, is that P squared?
Yeah, yes.
Really? That's homozygous dominant. But if you're dominant, does that mean you have to be homozygous dominant? No. What else could you be? Heterozygous. So this one's false. That's why we never start with how many people are dominant. We never start with that. What do we always start with? Recessive. How many are recessive? So if you know they're recessive, you know their genotype. If you know they're dominant, you don't know their genotype. They could be big A, they could be big A, little a. But if you know they're recessive, you know they're little a, little a. You know that. Okay, so 3 is true and 4 is false. Okay, class, I promised number 5, so you might want to star. I'm literally going to ask you, in Hardy-Weinberg, what symbol is the frequency of the recessive allele? It's Q. In Hardy-Weinberg, what symbol is percent heterozygous? 2PQ. How about in Hardy-Weinberg, what symbol is homozygous recessive? Q squared. In Hardy-Weinberg, what symbol is the frequency of the dominant allele? P. See what I'm talking about? Okay, I'm going to ask you each of those things. So know what each thing stands for. Yeah, it's definitely page 167. Given a set of genotypic frequencies, what would you expect the frequencies to be the next generation if the population obeys Hardy-Weinberg? If the population obeys Hardy-Weinberg, it means it's in equilibrium. What's equilibrium? Balance. So let's just pretend. Let's say that in generation 1, P is equal to 0.5 and Q is equal to 0.5. What would P and Q be equal to in generation 2 if it's obeying Hardy-Weinberg? 0.5 and 0.5. But it's okay. P would still be 0.5 and Q would still be 0.5. Okay? How about after 5 generations? Same. How about after 10? The same. That's what Hardy-Weinberg predicts, that no change unless there's some evolutionary agent working on the population. Okay, so I promised you a Hardy-Weinberg. You guys have your calculators? If not, I have one up here you can try. We did this at the beginning of lecture today, but in case you don't remember, you always start with what?
Q squared.
Then what?
Q.
Then what?
P.
Then what?
P squared.
And then what?
Q.
Okay, so you guys work on that. And then when you're finished, check your neighbor. Are running low on time. What did you guys get for Q squared? Huh? 0.06. Let's see, what was that? 25 divided by 400?
But don't we round?
To 2 decimal places?
Yeah,
It wouldn't be 0.1.
Rounded 2 decimal places, Gabriel?
Oh, sorry.
You're good? Okay. Good. All right. So everybody's okay on 0.06, 6%? Okay, now we're going to do Q by taking the square root of 0.06. What's the square root of 0.06? That's what I got too, 0.24. Again, we're going 2 places behind the decimal. All right, how do we get P if we know Q? Good. So I got 0.76 for P. Everybody's okay so far? All right, then we just have to square 0.76. 0.58? Okay. And then 2PQ. Point how many? You got 36? Okay, what percent of these fruit flies are homozygous dominant? 58%, that's right. What percent of these fruit flies are homozygous recessive? 6%. And what percent of these fruit flies are heterozygous? 36. Everybody's okay on that? Super. Okay, what's the difference between founder effect and bottleneck? Well, let me describe one and you tell me what it is. So I had this island out in the bay. You remember I was telling you about the bay the other day? How they dredged out the bay to make the ship channel? Well, you know, when they dredged out the bay, that was dirt they dredged out. They just piled it up out there and they made islands. There's islands out there. I don't know if you've been out there, but they're called dredge islands. Okay, so there was this dredge island that was just a pile of dirt, and these two brave, very brave lizards hopped on a piece of wood in Shore Acres, and they floated over to that little island to start a new colony of lizards. That's brave. What do you call that? Founder effect. Well, they liked it out there real well. There's lots of insects, places to hide. They multiplied. They got all these lizards. But old-timers talk about way back there was a big freeze. The old-timers said it got so cold, I'm not making this up, they said it's true. You could walk from LaPorte to Baytown. Walk across the bay. That's cold, people. That's just what they say. Anyway, it got so cold that it killed off most, but not all, of the lizards. What do you call that? Okay, that's a bottleneck. Okay, on the difference between founder effect and bottleneck. Okay, great. All right, lastly, I need some examples of natural selection pressures. I'm going to give you an example. Nobody ever thinks of this one. Disease. Disease kills off a lot of animals, a lot of plants. You can even think about humans. If you have resistance against disease and the disease comes, then you're good. Otherwise, you're not. Okay, give me something else. What else? That would cause natural selection. I got disease.
The environment.
Okay, we'll do environment, like too cold, too hot, too windy, too wet.
How would you say, like, lack of food
Okay,
or resources?
just resources. I'm going to go drought and environment. Ooh, this one's a good one.
Predators.
Yeah, that'll cause natural selection. Predators. Now, if you had natural selection on a population, what would that do to Hardy Weinberg? Would it change it? Oh, yeah. Natural selection will definitely change your P's and your Q's. Everybody okay on that? All right, great. So I've recorded this.